My name is Phoebe Strell, and I'm a PhD student at the University of Minnesota. Today, I'm going to talk to you about my work on generating microglia-like cells for Alzheimer's disease. You probably know someone that is directly or indirectly impacted by Alzheimer's disease. As of 2020, it impacted 55 million people worldwide, with numbers projected to double every 20 years. Even within the US, as of 2022, there are approximately 6.5 million Americans ages 65 years and older with Alzheimer's disease. This means that about 1 in 9 people in that population has Alzheimer's disease. This number is only expected to increase with the growing elderly population. Alzheimer's disease is one of the driving causes of dementia, making up 60 to 80% of all cases. Changes begin in the brain much earlier than the onset of the symptoms that we often see in clinics. Alzheimer's disease symptoms start with more mild cognitive impairments early in the disease and progress to cases of more severe dementia. Affected individuals can experience impairments in higher order functions, like struggling to plan or problem solve. They may lack awareness with things like time and location. They can have difficulties performing everyday tasks, and they can even have changes in mood, like anxiety and depression levels changing. Along with the cognitive decline, we see changes in the Alzheimer's disease brain. Some of the changes we will see include accumulation of protein that form plaques or tangles, the eventual damage and loss of neurons, and in recent years, dysfunctional microglia have been implicated in Alzheimer's disease. So, what do microglia do in Alzheimer's disease? Microglia play roles in removing small fragments of protein and corralling off plaques to minimize any damage to surrounding neurons. They also release inflammatory factors to surrounding cells and make reservoirs of these toxic proteins that lead to further neuronal damage. Microglia dysfunction can further drive the neurodegenerative processes we see in Alzheimer's disease. We see marked changes in the expressions of hundreds of genes in Alzheimer's disease brains. Even microglia found close to plaques have changes in their gene expression, as well as their ability to remove protein or corral off plaques. Dysfunctional microglia can secrete factors such as inflammatory mediators, leading to a chronic inflammatory environment within the brain. Finally, dysfunctional microglia may activate part of the immune system that results in synaptic overpruning, which inappropriately engulfs and prunes existing synapses that result in the disconnection of neural circuits important for retrieving previously established memories. Because microglia contribute to Alzheimer's disease, they are a reasonable cell to target for treating the disease. My lab is using blastocyst complementation to produce healthy exogenic microglia for treatment of Alzheimer's disease. Blastocyst complementation allows us to take donor stem cells, like induced pluripotent stem cells, from a mouse and inject them into a genetically modified blastocyst, which is an early-stage embryo. The genetic modification is made by using the CRISPR-Cas9 system, which makes a gene non-functional by inserting or deleting a base pair into the genetic code. By targeting a gene important for microglial development, we want to create a developmental niche for microglia. Our injected donor stem cells can then fill that niche and use the cues and signals from the mouse to grow and develop into functioning healthy microglia. We are currently exploring this Alzheimer's disease therapeutic of generating exogenic microglia-like cells in rodent models, with the eventual goal being to use human donor stem cells in a large animal host such as a pig to generate microglia-like cells that are specific to an affected individual. And with that, I would like to thank the researchers within the Low Lab and the University of Minnesota that have made this work possible.